आज चाहिँ हामी हिम सिन्थेसिसको बारेमा चाहिँ कुरा गर्दै जान्छौँ नि। हिम भनेको चाहिँ वान अफ द इम्पोर्टेन्ट मोलिक्युल हो, किनभने चाहिँ हामीले जस्तै कति प्रोटिनहरू हुन्छ, जस्तो इम्पोर्टेन्ट त हिमोग्लोबिनहरू भयो, मायोग्लोबिन हो, यसमा चाहिँ एकदमै एकदमै इम्पोर्टेन्ट रोल प्ले गरेर हुन्छ है यसको सिन्थेसिसहरूको लागि चाहिँ। सो हिम सिन्थेसिस अकर इन अल म्यामल टिस्यु, मोस्टली इन बोन म्यारो एन्ड लिभर। जस्तै इन बोन म्यारो फर सिन्थेसिस अफ हिमोग्लोबिन एन्ड इन लिभर फर सिन्थेसिस अफ साइटोक्रोम्स। Heme is a metalloporphyrin, must consist of a porphyrin and a metallic ion. It is a planar molecule, one of the most stable compound. First, knowing about the structure of heme or any porphyrins, we should know the structure of pyrrole. So this is the structure of pyrrole ring. See, porphyrin is a cyclic compound. It is formed by the linkage of 4 pyrrole rings to methylene bridges. You can see here 1, 2, 3, 4. These are the 4 pyrrole rings, and the one which is highlighted here is the methylene bridge that helps to connect the pyrrole rings. Methylene bridge, methylene bridge, methylene bridge. This is the basic structure of porphyrin. So porphyrin is mostly characterized by feature of it has a feature to form complex with metal ions that is bound to nitrogen atom of pyrrole ring. If it forms complex with iron, then it is known as heme of hemoglobin. If it forms complex with magnesium, that is known as chlorophyll, which gives pigment or color to plants. The 3 most important porphyrins are uroporphyrin, coproporphyrin, and protoporphyrins. These all are porphyrins, but they are named differently because they have got different side chains in porphyrin. Most water-soluble porphyrin was asked. It is uroporphyrin, and least water-soluble porphyrin is protoporphyrin. So this is the representation of porphyrin on the basis of Fisher's shorthand representation. This 1, 2, 3, 4 are pyrrole rings. This Roman 1, 2, 3, 4, and this numerical 1, 2, 3, 4, 5, 6, 7, 8 are the position of side chains. There are various side chains in porphyrin. Some of them are methyl, acetate, vinyl, and propionate. So we're going to talk about the structure of the 3 most important porphyrin. Uroporphyrin has acetate propionate side chain. In coproporphyrin, acetate is replaced by methyl side chain, propionic, methyl propionate. In case of protoporphyrin, there is methyl vinyl, methyl propionate, and methyl propionate. If this forms complex with ferrous iron, then it is called heme. You can see here, this is the structure of heme. So this is the important heme containing proteins with its functions. Now biosynthesis of heme. Site in almost all tissue in the body except mature erythrocyte. 85% of it is formed in erythroid precursor cell in bone marrow, and remaining is formed in hepatocytes for the formation of cytochromes. Starting material is succinyl-CoA and glycine, and it happens in mitochondria. We'll explain it. This is the basic steps. We'll go step by explanation. In step 1, there is condensation reaction between succinyl-CoA and glycine to form alpha amino beta ketoadiptic acid, which later is decarboxylated by the same enzyme to form alpha amino levinate or delta amino levinate, also known as delta ALA. Site of first step is mitochondria. First step is rate limiting step, and cofactor is also involved in this reaction. You can see it in here. The cofactor involved in this reaction is vitamin B6. This happened in mitochondria. So 8 molecules are condensed and 8 molecules of delta ALA is formed. Now 2 molecules of delta ALA is condensed to form 1 molecule of porphobilinogen and 2 molecules of water. So 8 molecule of delta ALA will form 4 molecule of porphobilinogen, also known as PBG, and this step is catalyzed by an enzyme alpha ALA dehydratase. This ALA dehydratase. This occurs in cytoplasm. Now most of the step until the formation of coproporphyrinogen of 3 occurs in cytoplasm. So ALA dehydratase catalyzes this reaction. And you should know that the first precursor pyrrole is PVG. This is the first precursor of pyrrole. The enzyme involved is ALA dehydratase, already told. We should know that ALA dehydratase is zinc containing enzyme. This enzyme is sensitive to the inhibition of lead and it can be inhibited in condition of lead poisoning. Now step 3 is 4 molecule of PBG is condensed in head to tail manner to form linear tetrapyrrole hydroxymethylbilane. In this reaction 4 molecule of ammonia is released, take place in cytoplasm and the enzyme involved is uroporphyrinogen 1 synthase, also known as PBG deaminase or HMB synthase. Step 4 there is formation of uroporphyrinogen 3 and uroporphyrinogen 1. The enzyme involved is uroporphyrinogen 3 synthase. In this case, the hydroxymethylbilane HMB is a linear tetrapyrrole which has two fates. One, due to spontaneous cyclization, it can form uroporphyrinogen 1. This occurs in certain porphyria. And due to action of uroporphyrinogen 3 synthase, it can convert into uroporphyrinogen 3. Step 6 is formation of coproporphyrinogen 3 from uroporphyrinogen 3. This is a decarboxylation reaction. 4CO2 is released. Enzyme involved with uroporphyrinogen decarboxylase. This can also convert the uroporphyrinogen 1 to coproporphyrinogen 1. So it can catalyze the formation of coproporphyrinogen 1 from this intermediate also. Now the site is cytosol. Now there is formation of coproporphyrinogen 3. Now it enters mitochondria. In mitochondria there is formation of protoporphyrinogen 9 from coproporphyrinogen 3. The enzyme involved is coproporphyrinogen 3 oxidase. The site is mitochondria. This enzyme is able to act only on type 3 coproporphyrinogen, so it will not act on this type, coproporphyrinogen 1. Step 7 there is formation of protoporphyrinogen from 9. There is formation of protoporphyrin 9 from protoporphyrinogen 9. This enzyme is protoporphyrinogen oxidase and this also occurs in mitochondria. Now the last step is formation of heme by incorporation of iron. This is the final step. Iron is incorporated into protoporphyrin. The enzyme involved is ferrochelatase, also known as heme synthase. It takes place in mitochondria and this enzyme is also inhibited by lead poisoning. Now the rate limitation of hemes or the regulation of heme synthase. The first step catalyzed by ALA synthase is the rate limiting step. I already told you there are two forms, ALA1 and ALS1 and ALS2. ALS1 is known as hepatic form and is found mostly throughout the body, and ALS2 is known as erythroid form, found in erythroid precursor cell. Only ALS1 is regulated. Remember, ALS2 is not regulated by heme or induced by drugs. Regulation is shown below. The things or the what induce ALA synthase are alcohol, hypoxia, drugs like barbiturates and greaseprevin. What inhibit ALA synthase are glucose and heme. So it's all about heme synthesis. You should know the important intermediates and important reactions, rate limited reactions. And also there is one thing, deficiency of ALA synthase can lead to sideroblastic anemia, and ALA dehydrase and ferrochelatase are inhibited by lead poisoning. And other enzymes that are found in this process is also important, and their deficiency can lead to different conditions of porphyria that will be discussed later in different video. Thank you.